in those phases. So bacteria, it's a prokaryote, and it reproduces by binary fission, which is the process by which one parent cell it's divides to form two progeny cells, and one cell give rise to two cells. So we say that uh, there is exponential growth in the bacteria. So one bacterium when it give rise to two bacteria, and after like 4 cycles, it will produce 16 bacteria. So this is how fast the bacteria can grow. So you can see this is one bacterium, and it is divided into two bacteria, and in 30 minutes you can see that in 30 minutes that it has divided into two, and in like 60 minutes you can see it has divided into four. So in 1 hour one bacterium divides into four bacteria. In the same manner, there may be other bacteria in the colony, so you can imagine the amount of bacteria which can result in 24 hours. So this one bacterium, after like 120 minutes, it has given 16 bacteria. So if there is more than one bacteria, so you can imagine the number of bacteria after 4 divisions. So binary fission is the process by which a bacteria divides and grow by increasing in cell number, and replication is this is a method of replication by the bacteria and increases in population. So how is different from mitosis is that binary fission is it occurs in prokaryotes, whereas mitosis occurs in eukaryotes. And in binary fission, um, there is the bacteria is actually not growing in size, but it is replicating. Whereas in mitosis, this occurs when there is a growth requirement in an organism. So generation time is the time required for a population of a bacteria to double in number. And what does it depend on? It depends on what the species doubling, what is the amount of nutrients, whether that species or that bacteria is present, the temperature, the pH, and other environmental factors. E. coli doubles every 20 minutes, whereas Mycobacterium tuberculosis doubles every 12 to 24 hours. So Mycobacterium tuberculosis is very slow to double, and as compared to E. coli, which is very fast. Okay. Now there are specific phases of growth in bacteria. The first is the lag phase, and it is the log phase, stationary phase, and then the dead phase. So we will see what is the lag phase. Now early part of the growth is the lag phase in the phase of binary fission in a bacteria. Vigorous metabolic activity is there, but the cells do not divide. So bacteria, they are preparing to divide, and this can last for a few minutes to up to some hours. So if we draw a graph representing on the y-axis the number of cells, and the x-axis tells us the time. This is the time when we inoculated the bacteria on a culture media or any place where there is requirement of the nutrients are there in a good amount. So you can see this phase is a lag phase where the bacteria, there is high metabolic energy in the bacteria, but it is not dividing. And this phase is the lag phase, so the bacteria is getting ready to multiply. Now the log phase or the logarithmic phase is when rapid cell division occurs, and the bacteria, they're using nutrients and secreting waste products. Now in this log phase, the rate of growth of the bacteria is more than the rate of death. So the drugs like beta-lactam drugs such as penicillins, they target the bacteria in the log phase because the drugs are only effective when the cells are making peptidoglycans or when they are dividing. So you can see here that in the log phase, the number of cells are increasing, and in a very short period of time, you can see how much the number of cells are there. So this is the log phase in which there's an exponential growth of the bacteria. Now the third phase is stationary phase, when the nutrients are depleted or toxic products cause growth to slow until the number of new cells balance the number of cells that die, resulting in a steady state. So in this stationary phase, the rate of growth is equal to the rate of death. So here you can see the same graph where the number of bacteria is not increasing and it is not even decreasing. So this is the stationary phase when the rate of growth in the bacteria is equal to the rate of death, and this occurs when the nutrients are getting depleted. Now the death phase is the final phase, which is marked by a decline in the number of viable bacteria, in which the rate of growth is less than the rate of death. So more of the bacteria are dying in the death phase, and you can see the number of bacterial cells, they are declining and going towards the baseline. Now this is another diagram which shows this is from your Levinson, and this gives the phases of bacteria. The A is the lag phase, then B is the log phase, then C is the stationary phase, and D is the death phase. So if in auspice you're asked what is the C phase, what is the phase which is marked by C, so you can tell the examiner that it is a stationary phase, but you have to study the logarithmic number of cells and the time, and then you tell the examiner. Okay, now we come to the aerobic and anaerobic growth in the bacteria. An adequate supply of oxygen enhances the metabolism and growth of the bacteria. Oxygen is important in the final steps of energy production. Use of oxygen generates two toxic molecules, hydrogen peroxide and the free radical superoxide. Now bacteria, they have two enzymes, superoxide dismutase and catalase. Now the superoxide dismutase is capturing this oxygen radical, converting it into hydrogen peroxide, and the catalase is then breaking the hydrogen peroxide into water and oxygen. Now obligate aerobes, these bacteria, they require oxygen. Facultative anaerobes, they utilize oxygen, but they can use the fermentation pathway to synthesize ATP in the absence of sufficient oxygen. So facultative anaerobes, as the name say, okay, they the first choice of these bacteria is oxygen, but if it is not present, then they can utilize the fermentation pathway. Obligate anaerobes, that means they cannot grow in the presence of oxygen because they lack either superoxide dismutase or catalase or both, so they die if they're exposed to oxygen, because these enzymes, superoxide dismutase and catalase, is absent in the anaerobes. Aerobic bacteria, the most important bacteria, Mycobacterium tuberculosis, it requires oxygen, uses all these pathways of energy generation. They do not have a mitochondria, but they use the plasma membrane. Facultative, they use oxygen, but they do not require oxygen. They can use the fermentation pathway to generate energy, for example E. coli. Obligate anaerobes, they do not require oxygen, and it is toxic to these bacteria like Clostridium. Aerotolerant anaerobes, they do not require oxygen, but oxygen do not cause them harm like Lactobacillus. Microaerophils, they require oxygen less than the atmospheric oxygen, for example Campylobacter jejuni. So here you can see the anaerobes, because they do not, when the oxygen is present, all the oxygen radicals are there, they do not have the superoxide dismutase and catalase, so it results in bacterial death. But whereas in aerobes and facultative, these enzymes are present and they can convert the toxic radicals into non-toxic products. Okay, so obligate aerobes, they are put into a test tube containing media. So you can see that obligate aerobes, they require they are mostly they grow where oxygen is present. Facultative, they can grow where oxygen is present as well as where the concentration of oxygen is low. Obligate anaerobes, they require oxygen free environment, so mostly if they are inoculated in a media, so they occupy the base of the test tube. Microaerophils, they do not require oxygen, and it is not even toxic to them. Aerotolerant is this oxygen does not harm them, and they do not require oxygen. But microaerophils, they require a little bit of oxygen. Now, fermentation of sugars is a metabolic process that consumes sugars and generate energy. The term fermentation refers to the breakdown of sugars like glucose and maltose to pyruvic acid and then to lactic acid. E. coli can be differentiated from Salmonella and Shigella on the basis of fermentation of lactose. Lactose is a disaccharide composed of glucose and galactose, and fermentation is also known as glycolytic pathway, and this is the process by which facultative bacteria generates ATP in the absence of oxygen. Now, when the bacteria fermentates glucose, it is converted into pyruvate and lactase, and it turns the medium. It changes the color of the medium according to the indicator. For example, if the phenol red is present as an indicator, the medium changes yellow. I showed you the picture of different medias, culture medias, and when we discussed triple sugar iron media, you can see that it was red in color, and then if there was fermentation reaction in the media, the color of the media changed into yellow. This is the same picture I discussed with you today in the practical of culture media. So there's fermentation which is occurring in this media, and it has converted from one color to another. That means fermentation process has occurred. Now, iron metabolism, bacteria, for its growth, it requires iron, um, uh, and they have these bacteria, they have a very high iron binding compounds called siderophores, which are required by these bacteria to grow. Enterobacter produced by E. coli is secreted by the bacteria. They capture the iron by chelating it, and then attached to specific receptors on the bacterial surface and are actively transported into a cell where the iron becomes available for use. Okay, now I will discuss with you normal flora of skin. Now, human body contains 10 raised to the power 13, that is more than a trillion cells, and more than a trillion quadrillion जो cells हैं, बैक्टीरिया हैं ह्यूमन बॉडी में. Cells human body के they are more than trillion, and quadrillion bacteria हैं which are occupying numerous places in our body. Now, the nose, mouth, pharynx, and larynx and trachea, they have these bacteria, Streptococci, Neisseria, Corynebacterium. The skin, it may have Staphylococci and Corynebacterium. Intestine, it may have bacteroids, Proteus, Pseudomonas, Bacillus, Clostridium, and Streptococcus. Parasites are those bacteria that live in association with a living thing, and they are dependent on them for nutrients and a suitable environment. Saprophytes